Hello everybody, welcome to session 5 of our lab video series. This lab session 5 focuses on collagen assay. This is for the course Cell and Tissue Engineering. So in this lab, you can see that on the picture here we have a Circol soluble collagen assay kit. This is a commercially available kit that's available from the company BioColor, and we're going to be using this kit to determine the soluble collagen content within our gel samples. Recall that in lab session 3, we made those cellular gels consisting of mesenchymal stem cells that were seeded within the agarose gel. And over the period of culture, those cells presumably produced collagen, and now we're going to measure how much collagen was produced. So when you open the Circol soluble collagen assay kit, you'll find several items within the kit, namely a dye reagent, an alkali reagent, a reference standard, an acid salt wash reagent, an acid neutralizing reagent, and last but not least, a collagen isolation and concentration reagent. Two items in particular I want to point out. One is the dye reagent, which contains that all-important serious red dye, which is used to target collagen within your sample, and it binds to it. And when it does, that can be detected using a spectrophotometer, and the absorbance can be red. The second thing is, for that absorbance to be converted into a collagen content, we would need a standard curve between absorbances and collagen of known concentrations. And so for that purpose, we have the item number 3 here, which is the reference standard, which essentially allows us to dilute it and build a plot of absorbances with known collagen concentrations. And so then when we have the absorbances for our samples, we can refer to this linear plot of collagen versus absorbances, and then we can then convert our absorbances into collagen content using that plot to convert absorbances of the measured dye into collagen content, since the dye specifically binds to collagen. So besides what is supplied within the Circol soluble collagen assay kit, we also need some additional items to conduct this collagen assay. Those are namely acetic acid, pepsin, conical microcentrifuge or Eppendorf tubes, and deionized water. I want to point out that pepsin is used to digest the collagen, and so when we have collagen within our samples, we need to make sure that it is digested completely, and the soluble component can then be measured using absorbance with a spectrophotometer. And there are other agents that can also digest collagen, such as the enzyme collagenase, but pepsin in particular has been recommended with the Circol assay kit, and so that's what we're going to use to digest the soluble collagen. In addition, just to point out, we have deionized water, and we're going to be using deionized water to dilute the known collagen standard that's provided in the kit, so that we can essentially have collagen content of known values from more dilute to less dilute, and we can then read the absorbances of those samples and then build our standard collagen curve with respect to absorbance. And of course, once we have that, we can use that standard curve to convert our sample absorbances into collagen content. So now we can go ahead and prepare our collagen standards. So here we have 3 empty Eppendorf tubes, and we're gonna start with these, and we're gonna label them as S1, S2, and S3, as you can see here. And we're simply going to pour the following volumes into S1, S2, and S3 respectively of the collagen standard that came in the kit in the volumes of 10, 20, and 30 microliters respectively. So next, we want to fill up the Eppendorf tubes S1, S2, and S3 with an equal volume of solution. And so in order to do that, we're going to fill up tubes S1, S2, and S3 with 90, 80, and 70 microliters of deionized water or DI water respectively. So when we do that, recall that S1 had 10 microliters of known collagen solution versus S3 had 30 microliters of known collagen solution. And so when we added 90 and 70 microliters respectively to tubes S1 and S3, they both have 100 microliters of solution, of collagen solution, but clearly the concentration of collagen in S3 is higher than what is there in S1. And so in this manner, we can build our known collagen concentrations, and once we measure the absorbances of these concentrations, then we can build our collagen standard curve. Once we have our collagen standard curve, then we can use it to then convert our absorbances for our samples into collagen content for our gel samples. So next, we're going to also prepare a collagen blank sample, which essentially has no collagen in it. So it's 100% deionized water or DI water, and this will just simply be labeled as S0. So next we go to our sample preparation, our cellular gel sample preparation. So the first thing we need to do is to extract the collagen from the gels. As I mentioned, we're going to use pepsin to digest out the collagen into the solution. And so specifically, our pepsin digest solution to digest the collagen out of the gels and into the solution can be done doing the following. So we will have to make a gel digest mixture, and this will be done by preparing a pepsin concentration of 0.1 milligrams per ml in half molar acetic acid, and this digest mixture will be stored at 4 degrees centigrade. Now note that the agar gels that we have been culturing may have slightly different volumes, therefore we need to also measure the wet weight in grams of each of these gels. So keep in mind that if you're trying to report collagen content, the assumption is that all your gels are essentially the same, which we know they're not exactly the same. So one way we can normalize the collagen with respect to sample variation based on their weight or volume is to normalize it with respect to their weight. So instead of reporting the collagen content as 5 micrograms or 10 micrograms, it would be reported as collagen concentration per unit weight. So it'd be 5 micrograms per gram or 10 micrograms per microgram. So it would be normalized to the wet weight, and therefore we are comparing apples to apples. All right. So again, because the agar gels may be somewhat different in their sizing, we need to normalize them by measuring their wet weight before we start with the digest. Okay, so here we go. We measure the wet weight in grams of each agar gel. And once we have the wet weights recorded, then we can proceed with the pepsin digest. And when we eventually get our collagen concentrations, we can normalize them with respect to that sample's wet weight. So, if the concentration is microgram per ml, then it would be actually microgram per ml per gram, so per gram wet weight. So therefore we are comparing apples to apples when we are comparing between samples and between our groups of 0, 10, and 30% FBS. So now that we have measured the wet weights of our samples, we can go ahead and proceed with our digest, with our prepared digest solution. And so we will digest our cellular agar gels with the 0.1 milligram per ml half molar acid pepsin digest mixture that we prepared. And this will be occurring within an ice water container overnight. So the digest will happen overnight at roughly 4 degrees centigrade within this ice water container. And we're just going to leave it to digest overnight, which is about 8 hours. 8 to 12 hours, let's say, roughly. So the next day, next we're going to add 100 microliter of acid neutralizing reagent to every 1 ml of extract, as well as add 200 microliter of cold isolation and concentration reagent. The reason for doing this, as the name implies, is to neutralize the chemical reaction process. We are done with our extraction of the collagen, and we want to stop any further reactions that may occur within these tubes. And to enhance the process of neutralization further, we can then also mix the tubes well by inverting the tube or by vortexing the tube using an agitator. And another step that would be good to do, just to be sure that we have really neutralized everything, is to then place the tubes, the Eppendorf tubes, for another overnight stay within our ice water container at 4 degrees C. And this is just an extra safety step to make sure that all our collagen solution and the chemical process within each tube has indeed been neutralized. Again, overnight being anywhere from 8 to 12 hours, roughly. So then the next day, the tubes containing our gel digest mixture now will be centrifuged at 8,500 RPM for 10 minutes. We can safely then, at this point, discard the supernatant without disturbing that valuable pellet at the bottom of the tube. As can be seen here, the supernatant can be discarded. The sample tubes will be labeled according to the corresponding conditioning groups. There should be an N of 3 samples for each of our groups, being the 0, 10, and 30% FBS groups, the 3 groups, and we label them accordingly as seen on this slide. For example, 0% FBS or T1, T2, T3, and so on. Next, we will add the serious red dye that came in our Circle assay kit, and we will add 1 ml of the dye reagent to all the samples and standards. So 1 ml of the dye for each sample, or to each tube in which we have a sample, and to each tube in which we have a standard. Okay, so 1 ml of the dye reagent will be added to each sample and to each standard tube, and this will then be placed on a shaker for 30 minutes to ensure that there's good mixing between our dye reagent and our sample, or between our dye reagent and our standards. We're going to do a centrifuge step again. Similar to what we did before, we're going to centrifuge our tubes at 8,500 RPM for 10 minutes. And once again, the supernatant will be carefully discarded without now disturbing that pellet at the bottom, which this time will be our soluble collagen pellet, okay, or a pellet that contains soluble collagen that was essentially secreted by our mesenchymal stem cells while they were residing in our agarose gels in culture. Next, we will add 750 microliter of ice cold acid salt wash reagent to the pellet, to each of these tubes. Again, with our centrifuge step, so after we've added that acid salt wash reagent and mixed it in, we're now going to centrifuge at the 8,500 RPM for 10 minutes once again. The supernatant again will be discarded, and we will have our cleaned up collagen pellet at the bottom of each of these Eppendorf tubes. Next, we will add 300 microliter of alkali reagent to the pellet in each tube. The mixture will be vortexed until the pellet has completely dissolved within the reagent. Okay, as you can see here, we can also enhance the mixing using a shaker or a vortex, and we will continue to shake or vortex the samples until the pellet can no longer be seen and has completely dissolved within the alkali reagent. At this stage, we are getting really close to starting our absorbance reading measurements to read our assay results. So we're basically going to first take the alkali reagent mixture and position it in triplicates into a 96 well microplate at a volume of 100 microliters per well. So basically we have a triplicate in terms of readings. We've got 3 readings per sample, and that's just to avoid errors that may be caused by uneven mixing or odd samples. There may be some outliers. So we try to get 3 readings per sample, and so we basically pipette the alkali reagent mixture in triplicates for each sample, okay, per sample into a 96 well microplate, and again the volume that we use is 100 microliters per well. Okay. Next, we are ready to measure the absorbances of our samples, and so to do that, we typically use an instrument known as a spectrophotometer, or also another instrument is known as a microplate reader. And so we place our micro well plate into a microplate reader, such as what is shown on this slide. And then this instrument allows us to specify the wavelength in the nanometer range. And as recommended by the manufacturer specifications, in other words, from BioColor for the collagen assay kit that we're using, the recommended wavelength for the Sirius red dye that binds to the collagen is 555 nanometer. So we specify that wavelength with the instrument, and then we can subsequently proceed to determine the absorbances of our collagen solutions within our samples, or derived from our samples, as well as from the standards that we prepared. So now that we have measured the absorbances for all our samples as well as our known collagen standards, we can then proceed to plot the data of our known samples S0, S1, S2, and S3 with their corresponding measured absorbances, which we just did with a microplate reader for our collagen known standards as well as for our samples themselves. And so here we have the data points for the corresponding absorbances for our 4 known concentrations of collagen, namely S0, S1, S2, and S3. And from this data, we can then proceed to determine the best linear fit equation for these known collagen standards. And so now once we have this equation, we can then go back to our samples for which we have absorbance values, but we don't have the collagen concentration. But if we plug in those absorbance values into this equation that we determined from this fit, then we can compute the corresponding collagen concentration for our samples based on their absorbances and based on the best linear fit equation derived from these known collagen standards. This concludes session 5 video. I'd like to greatly acknowledge doctoral student and our TA, Ms. Denise Su, for greatly assisting with this PowerPoint presentation. Corresponding data that you will need to analyze for this lab session will be posted onto Canvas shortly within our Cell and Tissue Engineering course Canvas shell. Again, this concludes session 5. Thank you very much.